Well, hello everyone. My name is Andrés Comba, and as he said, I work for Bioeconomy for Change. I'm a consultant, and Bioeconomy for Change is a bioeconomy cluster in France. And I'm happy to present you the following webinar that will focus on the following points: enzymes market and how enzymes are increasingly used for different type of applications, the barriers that biocatalysis present, and last but not least, how innovation is trying to address these challenges. Let's start by what are enzymes. Enzymes are biological catalysts and have a high specificity for their substrate. Enzymes are used in a wide variety of applications, such as agri-food, chemicals, cosmetics, for the production of detergents, paper or leather, for the production of biofuels like ethanol, pharmaceuticals, and for research and biotechnology. The use of enzymes have a lot link of benefits. We can say they reduce energy consumption, reduce production time, improve sustainability performance. They provide safer working conditions for workers. They reduce waste generation, and they offer better quality products. Let's see here the market. The global enzyme market, according to recent figures, was worth 10.6 billion in 2020, and it's expected to reach over 17 billion by 2027. This is growing a compound annual growth rate of over 6% during the following years, which is significant growth. Factors driving the growth of the use of enzymes are growing consumer awareness of processes that has less impact on the environment, search for products with better technical qualities and healthier, increases in process safety for workers, and reducing the use of raw materials and energy consumption in manufacturing processes. Enzymes can be classified by the type of chemical reaction they catalyze, such as hydrolysis, oxidoreductases, transferases, liases, and others. Among them, hydrolysis represent more than 50% of the enzymes market. This is because they can participate in a wide variety of biological processes, as they are the most numerous and diverse class of enzymes. The second most important type of enzymes are oxidoreductases, which cover around 20% of the market. Enzymes can also be classified by the type of substrate on which they act. As we can find, carbohydrates, proteases, lipases, nucleases, among other groups. Carbohydrates, proteases, and lipases dominate the enzyme market, accounting for more than 70% of the enzyme sales, as can be seen in the graph on the right. They are used in a wide variety of applications, mainly in the food and beverage industries, but also in detergents, animal feed, and many other applications. Carbohydrates cover a wide variety of enzymes involved in the hydrolysis and synthesis of carbohydrates, such as amylases, invertases, and glucosidases. Proteases represent the second largest group. They break the peptide bonds of proteins and are mainly used in food and beverage industry, in detergents, and in cosmetics. Within the enzyme market, we can differentiate between industrial and specialized enzymes. Industrial enzymes represent the largest market share in terms of volumes and values. Industrial enzymes have different uses, such as the manufacture of food or beverages, animal feed, detergents, the production of cosmetics or perfumes, or the production of biofuels. The demand for these markets is mainly driven by the demand for carbohydrates and proteases in food and beverage applications, mainly in China, India, and Japan. The nutraceutical sector in developed economies like Europe and North America. And agriculture, biofuels, and animal health and nutrition in Latin America. Let's look at some application of industrial enzymes, such as they are used in fight against plastic pollution. Carbios a French startup, has developed a biodegradation technology that uses optimized enzymes to recycle PET, a plastic commonly used in plastic bottles. PET is the most common polyester in the market, and approximately 7 million tons of PET are produced worldwide. These enzymes developed by Carrillos degrade PET into two monomers, purified therapeutic acid and monoethylene glycol, which are PET building blocks. Both monomers can be then recombined to recreate the food-grade PET polymer. These enzymes were optimized and can break down more than 90% of PET in less than 10 hours. The advantages of this enzymatic technology are reduction of water pollution, high process efficiency, as these enzymes have 100% recycling yield, and allow processing all types of PET plastics. And the polymerization is carried out under mill conditions for worker safety. One last innovative application of industrial enzymes is their use in agriculture to control pests. Enzyme-based biocontrol technology is an alternative to chemical pesticides. It effectively controls microbial pests, insects, and weeds. Rhizophoric microorganisms are first isolated from various cultures, then purified, and then lytic enzymes are obtained from biological control agents such as bacteria or fungi. Among these lytic enzymes, chitinases, cellulases, proteases, and glucanases have the function of limiting the invasion of pathogens into plants. These enzymes do not only eliminate pests and protect the crop, but are easily biodegradable. They provide high control efficiency as they act quickly, and they provide healthier agriculture products. One example is Enzymes, which is working with FMC, which is a large agriculture chemical company, on an enzyme-based technology to jointly develop a diamide insecticide as a fungicide, and a fungicide that can control Asian soybean rust, a major fungal disease of soybean that is problematic in South America. On the other hand, there is the market for specialty enzymes, which are used in smaller quantities, but for higher value-added products. This specialty market is already growing very fast. In 2012, it was worth $1.5 billion. By 2020, it had reached a value of $0.4 billion and is expected to grow at a compound annual growth rate of 7 to 8% in the coming year, reaching a value of more than $8 billion by 2028. Specialty enzymes will primarily drive enzyme demand in the coming years. Special applications market for enzymes include the pharmaceutical industry. For example, in drug formulations for the treatment of cancer and cardiovascular diseases. The pharmaceutical sector is the largest area of application for specialty enzymes. Another special application market is research and biotechnology. For example, in biological research for DNA amplification and sequencing. Enzymes can also be used to diagnose different types of diseases, such as COVID-19. Another use of enzyme is in biosynthesis for various types of chemical reactions. For example, the European project INCITE focuses on the solvent-free synthesis of natural oleochemical esters using lipase enzymes with applications in cosmetic and food industry. Another route of this project focuses on an esterase catalase production of a key molecule used as a starting material with applications in crop protection and public health. Let's look at an application of enzymes in a special market. In this case, Limonene, whose derivatives can have numerous applications, such as in food, cosmetics, pharmaceuticals, and agrochemicals. Limonene can be produced by microbial production, involving the use of the enzyme limonene synthase, which is an enzyme found in plants and convert genadyl diphosphate in limonene. This pathway is deployed indigenously in Escherichia coli, which hosts the enzyme limonene synthase. Other microorganisms, such as yeast or cyanobacteria, can be hosts for limonene production. What are the advantages of this biocatalysis pathway? Microbial limonene synthesis is characterized by being enantiospecific, thus providing a higher quality product. This is necessary for application in certain industries, such as in aroma and fragrance products or for certain food products. In addition, biotechnology production of limonene by microorganisms could allow its use in large volumes, such as in bio-based solvents, biomaterials, or fuels. Finally, it also reduces dependence on agricultural raw materials, which availability may vary throughout the year or from year to year. Let's look at who are the main players in the enzyme market. The enzyme market consists of a small number of large companies with the largest market share, which can be called an oligopoly, but which is highly competitive. No enzymes, Dow DuPont, DSM, or BASF are the most important players in this market. This map shows the most relevant geographical market for enzymes. North America represents the largest market, accounting approximately 30% of the global market revenue in 2020. Pharmaceuticals, food and beverages, personal care and cosmetics are major industries that demand large quantities of enzymes in this region. Europe is the second market, with enzymes applications in industries such as pharmaceutical and cosmetics, which are highly developed and require large quantities of enzymes. Finally, the Asia-Pacific region is characterized by an important growing demand in food and beverage applications, and in the pharmaceutical industry, especially in China, India, and Japan. This region is already becoming the second largest consumer market for enzymes. In this part, I will cover the challenge of enzymes utilization and how innovation is addressing these limitations. Let's begin by mentioning the challenges that exist when using enzymes. First of all, these three factors can destabilize the enzyme's activity. Enzymes are sensitive to pH changes and work best in narrow ranges. Therefore, pH control is often a critical parameter to consider in many biocatalytic reactions. Enzymes are effective at specific temperatures. Temperature differences may cause changes in reaction kinetics, product yield, or other critical process variables. Finally, enzymes usually operate in an aqueous medium, but many enzymes find it difficult to react in organic solvents that are often necessary to solubilize the reacting substrate, because organic solvents inactivate the enzyme and reduce its stability. Enzymes with high substrate specificity can be problematic if they do not accept the substrate. This means that they have little affinity for compounds with structure distant from their natural substrates. Moreover, in some cases, yield can be lower than those obtained by chemical reactions, and reactions may take longer. Cofactors. Cofactors are considered auxiliary molecules that help enzymes as activators. The regeneration of cofactor is a challenge, since in many biosynthetic pathways, enzymes depend on them, and cofactors are expensive. The industrialization of biotechnology using enzymes is therefore problematic, and it's difficult to find solutions that improve cofactor retention, or cheap and sustainable metal cofactors. The use of enzymes can sometimes make the process more expensive, or their availability is sometimes limited. In addition, their development time from research to use may also be long. Since natural enzymes may have limitations under certain conditions and applications, genetically modified enzymes are designed to improve enzymatic performance. But GMOs may pose risk, and so the debate about the potential problems of recombinant organisms arises. Biocatalysis has undergone a great development in the last 2 decades, as can be seen in this image, mainly as a direct consequence of remarkable advances in different disciplines. Among we can mention molecular biology, genetic engineering, and information technologies. Biocatalysis has great potential due to advances in genomic sequence extraction, gene synthesis, and computer-aided design, which create a much larger pool of available enzymes. The development of new enzymes is a breakthrough technology, and it involves the discovery or synthesis of new enzymes by using genomic sequencing and gene synthesis, combined with bioinformatics, to design enzymes for specific purposes. This is made possible by metagenomics, a technique that has emerged over the last 2 decades. This technique consists of analyzing the sequencing of the genomes of numerous individual organisms contained in genetic material extracted directly from environmental samples. What this disruptive technology allows is to directly isolate all the DNA present in the sample, be it soil, water, or other, rather than isolating microbes. In the past, enzyme discovery was done by collecting samples in nature and preparing microbial cultures in the laboratory, with the drawback that a low percentage of the microbes can be cultured, due to the limitation of the culture-based method. The metagenomics approach is one solution for researchers to overcome this problem. This approach uses methods such as gene construction, screening, gene expression, bioinformatic analysis, as well as structure prediction and characterization of protein products. Let's take a look at the image on the right. Microbial screening offers the possibility to exploit enzymes from unusual environments, such as extremophile enzymes with optimal growth in extreme environment conditions, like thermal waters, alkaline soils, or industrial effluents. Importantly, expansion of the enzymes pool will allow access to new enzymes with the desired activity to perform chemical conversions that are challenging or impossible using traditional organic chemistry, increasing energy efficiency, and reducing the environmental impact of production processes. The second point of improvement in biocatalysis is the progress made in the development of directed evolution through genetic engineering techniques, such as protein engineering. This is leading to increased enzyme efficiency, which will enable the deployment of biotechnology as well. To improve enzyme performance, protein engineering is used as a method. This innovation has 2 main methods that are not mutually exclusive: rational protein design on the left and direct evolution on the right. In the past, protein engineering was largely guided by structural and mechanistic information, also called rational protein design, to make the desired changes. But since the 90s, the use of protein engineering through selective evolution, such as DNA shuffling, has been of paramount importance for the deployment of industrial biocatalysis, enable dramatic improvement in enzymes properties, and thus cost-effective production. DNA shuffling is an in vitro recombination method that generates mutations in enzymes through random fragmentation and reassembling of genes. This results in the increase of the DNA library. These enzymes are then tested, and those that are more efficient at catalyzing the desired chemical reaction are selected. New random mutations can be reintroduced into the genes of the selected enzymes, working as a cycle. Today, enzyme engineering strategies can benefit from computer-aided design, which through algorithms and machine learning helps to identify the optimal amino acid sequences for certain functions, and thus modify existing proteins, improving their performance. Thanks to DNA shuffling, properties such as enzyme activity and stability can be increased, and antioselectivity improved. The range of enzyme substrate can also be extended. Through directed evolution, the ideal parameters for a process can be predefined, and then biocatalysis can evolve to achieve them. The advance in enzyme immobilization is an innovation which enables not only better catalytic performance under industrial conditions, but lower production costs. As enzymes are soluble in water, their recovery from aqueous effluents is often not easy, which make their single-use expensive and ineffective. Enzyme immobilization is a technical process in which isolated enzymes are bonded to a prefabricated carrier or support, such as synthetic polymer, a biopolymer, an organic resin, or inorganic solid. This immobilized biocatalysis can be enzymes or whole cells, and the attachment to the support can be by adsorption, ionic, or covalent bonding. Other methods of immobilization may be entrapment or cross-linking methods. Enzyme immobilization is done after enzyme enhancement by protein engineering, and is used to facilitate enzyme recovery and recycling by centrifugation or filtration, thus reducing the biocatalysis production costs. In addition, they often result in greater process stability and simplicity, and improved product quality as the product is not contaminated by the enzyme, which is useful in the food and pharmaceutical industries. Moreover, an immobilized system reduces the environmental footprint and provides greater tolerance to organic solvents by suppressing enzyme denaturation by protein unfolding. On the other hand, immobilization decreases enzyme activity due to dilution effects from the addition of non-catalytic materials such as the use of carriers, which generates interest in carrier-free immobilized enzymes. Therefore, cross-linked enzyme aggregation is a method of enzyme immobilization that combines two processes, immobilization and purification in one step, and the enzyme do not need to be of high purity. It should be noted that the demand of immobilized enzymes for industrial organic systems has been growing fast during the last years. Finally, advances in cascade biocatalysis will allow expanding the industrial application of biocatalysis. The aim of this technology is to carry out complex reactions in the form of sequential synthesis in one-pot operation, without the need for isolation of intermediates. The enzyme cascade processes can be carried out as isolated enzymes or whole cell systems, or by fusing genes from different enzymes to produce a single polypeptide with multiple enzymatic activities, a so-called chimeric enzyme. The multi-enzyme biocatalysis approach brings advantages such as avoiding several separation and purification steps, thus reducing unit operations, the use of fewer solvents, smaller reactor volumes, and shorter cycle times, to displace unfavorable reaction equilibria and simplify cofactor regeneration, and to increase yield and reduce waste. Some examples of cascade process involve their application for cofactor regeneration. What conclusions can be shared with you after this webinar? First, the enzyme market will continue to grow due to increasing consumer awareness of environmental and health concerns, and for research of products with improved properties. Future growth in enzymes demand will be driven primarily by special applications, and Asia will experience a very significant growth in enzyme consumption. The chemical industry has great challenges to face in the coming years, and biocatalysis is a technology that can provide a number of solutions while respecting the principles of green chemistry. Biocatalysis presents a number of challenges, such as the instability of enzymes or their high cost, their recycling, as well as the regeneration of cofactors. But a number of innovations have been and are being developed to address these challenges, such as the discovery of or synthesis of new enzymes, the improvement in enzyme performance through protein engineering, the mobilization of enzymes to reduce costs, and cascade reactions that also reduce production costs and simplify the biocatalysis process. Some of the objectives that biocatalysis must achieve in order to expand its implementation and application include increasing enzyme activity and thermal stability, as well to improve their compatibility with solvents, and improving biocatalysis performance, not only in terms of reducing production costs, but also in its ability to catalyze a wider range of reactions, making it more versatile. Well, thanks for all your hearing me, and I'm open for your questions.